DNA fingerprinting. The technique developed to identify a person with the help of DNA restriction analysis is known as DNA profiling or DNA fingerprinting. The technique of DNA fingerprinting was first given by British geneticist Dr. Alex Jeffrey in 1984. About 99.9% of nucleotide sequence in all persons is same. Only some short sequences of nucleotides differ from person to person. In the population, every person shows unusual sequences of 20 to 100 base pairs which are repeated several times. They are termed as variable number of tandem repeats, VNTRS. So you can see over here, every person has different VNTRS. The length of the region having VNTRS is different in each individual and hence is the key factor in DNA profiling. Steps involved in DNA fingerprinting are isolation of DNA, restriction digestion, gel electrophoresis, sudden blotting, selection of DNA probe, hybridization, and photography. Isolation of DNA. The DNA must be recovered from the cells or tissues of the host. Small amount of tissue like blood, hair roots, skin is required. Restriction digestion. The isolated DNA is treated with restriction enzyme. The restriction enzyme cut the DNA into small fragments having variable lengths. This phenomenon is called restriction fragment length polymorphism, RFLP. Gel electrophoresis. The DNA samples are loaded for agarose gel electrophoresis under an electric influence. The DNA fragments which are negatively charged move to the positive pole. The movement of these fragments depends on length of the fragment. This results in formation of bands. Double-stranded DNA split into single-stranded DNA by alkali treatment. Sudden blotting. The separated DNA fragments are transferred to a nylon membrane or nitrocellulose filter paper by placing it over the gel and soaking them with filter paper overnight. Selection of DNA probe. A known sequence of single-stranded DNA is prepared. It is called DNA probe. DNA probe is obtained from organisms or prepared by cDNA preparation method. The DNA probe is labeled with radioactive isotopes. Hybridization. Probe DNA is added to the nitrocellulose filter paper containing the host DNA. The single-stranded DNA probe pairs with the complementary base sequence of the host DNA strand. As a result, DNA-DNA hybrids are formed on the nitrocellulose filter paper. Remaining single-stranded DNA probe fragments are washed off. Photography. The nitrocellulose filter paper is photographed on an X-ray film by autoradiography. The film is analyzed to determine the presence of hybrid DNA. Applications of DNA fingerprinting. In forensic science, DNA fingerprinting is used to solve problems of rape and some complicated murder cases. DNA fingerprinting is used to find out the biological father or mother or both of the child in case of disputed parentage. DNA fingerprinting is used in pedigree analysis in cat, dogs, horses, and humans.